let me start out with something that kind of puts the gut microbiome and the microbiome in general in perspective. If you look at this image, so for about 3.5 billion years, there were only microbes on the planet Earth, the dominant life form. And they had a long time to obviously learn to communicate with each other and with the environment. And during that time, they were able to accumulate a remarkable amount of knowledge related to communication and language. And we'll come back to this, why this is so important. Then about 500 million years ago, the first microbes came up with the idea to settle in these floating digestive tracts of the hydra, very primitive marine animal that had these nerve nets wrapped around it that regulated its peristalsis and its movements, very similar to today's enteric nervous system that we have in our gut. So this is stayed for about that design principle 500 years. And then later, and then the microbes, through a process called lateral gene transfer, transferred a lot of their information about neurotransmitters and signaling molecules to this enteric nervous system of really the first brain. And then later, with the development of the main brain, many of the same genes were transferred into the central nervous system. And that explains why we have a language that really lets the microbes communicate with our enteric nervous system, regulating gut function, but also with our brain, and also lets the brain communicate with our microbes. What the big question is, what did these microbes come up in 3.5 billion years in terms of communication molecules? So everybody knows 100 trillion microbes by numbers are present. About 60% of our entire organisms are microbial cells, 40% human cells. But much more interesting is the fact that there's about 20 million genes that these microbes have from that long evolutionary period, which is obviously several-fold higher than our 20,000 human genes. We don't know what most of these microbial genes do today. So I think over the next decade or so, we'll learn a lot about this, and many new concepts will arise what these genes encode and what signaling systems that they can influence. So when you think about today's presentation about early development of the nervous system, so nobody mentioned the microbiome. It's kind of interesting because 99% of the genes that play a role in development and drive us have not really been taken into account because they come from the microbes. How much impact this will have on our current understanding and theories of our bodies, and particularly of our brains, that will need to be that will need to be settled in the next decade or so. On the left side, you see what the microbes generally do. That's why they have evolved. So they break down a lot of digestive products that the mammalian or the animal GI tract cannot absorb. They break it down into smaller molecules. Short-chain fatty acids, you probably have heard about this, is one of this, like a currency. But there's hundreds of thousands of metabolites that they produce by this process. Again, the majority of which we do not know what they do. What's most interesting, if you're not somebody who's just interested in metabolism, what's most interesting is that many of these metabolites have the ability to communicate not just at the gut level, but to all organs of the body, as shown on the right side. The cardiovascular system, stress system, my interest, the enteric nervous system regulating GI function, and also to the brain and behavior. So over a very short period of time, and keep in mind this science is about 10 years old in terms of the brain-gut microbiome science, so really at its infancy to understand it, but already a lot has evolved in our conceptualization of this. In this sort of complicated-looking slide, I just want to emphasize a couple of points here. There's the brain on the top, there's the microbes on the left side, and there's what I like to call the gut connectome on the right side. What this means is it's the gut cells, it's the enteric nervous system, 150 million neurons. It's about 40% of our immune system located there, and it's the biggest part of our endocrine system, producing all these hormones. All these cells communicate with each other, and the microbes have a way to influence that communication. So what we know today from mostly studies in animals is that the brain can communicate with both the microbes directly and with the gut connectome. The microbes can communicate to the gut, but also through that process signal to the brain. And the microbes can also signal directly to the brain through some of these neurotransmitter molecules that they produce that match our own neurotransmitters and have homologous receptors in the central nervous system. Well, and I should say so you see these arrows, perturbation. So this is really where stress comes in. Stress can come in at the brain level, psychosocial stress, but also perturbation of the gut microbes through antibiotics or perturbation of the gut with inflammation, inflammatory bowel disease, and other processes. Anytime there's a perturbation of one of those interconnected components, the other components will respond to it. So this is a system that always acts together as really a system and not as individual parts. In the adult, so you've heard a lot about it, and it's kind of an amazing field, particularly if you're a scientist and you get invited to meetings about nutrition and the microbiome. So there's hundreds of unsubstantiated concepts and theories that people propose at these meetings. If you go on the internet, you can buy hundreds of probiotics, prebiotics. Everything is being proposed as bringing your gut microbiome back into balance. So I'll not be talking about this other than saying once the microbiome is established early on in life, there's three factors that play a major role in modulating its output, not necessarily the organization that's stable, but its output. One is the brain, stress and emotions. Another one, probably the most important one in adult life, is diet. And the third one is what you can call xenobiotics, which means in our world medicine, antibiotics, pesticides, and other chemicals that the microbes really have never experienced in evolution, but because of their intelligence, remember 20 million genes and their products, they're able to metabolize those into other compounds, just like the liver. So people have called this, it's really a second liver that can do this. So this is the main topic of this presentation. The most important events in the programming of the gut microbiome and the interactions with the nervous system are summarized here, and there's a lot of them. They start in pregnancy, seen at the lower three images of the pregnant mother. Illnesses of the mother, particularly in inflammatory processes during pregnancy. It's been shown that, for example, during flu outbreaks, there is a spike in neurodevelopmental diseases like autism. We think today that this is one of the factors. So health of the mother, medications that the mother takes, and particularly the diet that the mother takes during pregnancy. It's been found to be one of the most important influences on the microbiome of the infant, what diet the mother was on during pregnancy. Then the way the baby comes into the world, C-section versus vaginal delivery, that has received initially a lot of attention because it seemed like really a spectacular thing, and people have started to do this vaginal seeding. So if kids are born with a C-section, to put some vaginal fluid on the baby afterwards. It's actually turned out to be by itself not such a huge factor, because after about a year, the microbiome of those two modes of delivery are pretty much the same. Probably more important, breastfeeding versus formula feeding. There's molecules in breast milk, these so-called human milk oligosaccharides. They're very large molecules that cannot be absorbed by the infant. So they're not for nourishing the infant. They're targeting at the microbes, and they program the establishment of the microbial architecture. Those are influenced by the genes of the mother and by the diet of the mother. There's hundreds of these molecules, and each mother has a different set of these molecules. They're not present in formula. Despite all the efforts that Nestle has put into this, they've never been able to synthesize these molecules. So probably millions of children in the developing world have grown up without this early programming. Another very important factor, I mentioned the antibiotics in the adult. They're much less important in the adult because you can treat somebody 3 times, so somebody can be on long-term antibiotics, and it will not fundamentally change the microbial composition unless you are unfortunate and develop a process called C. difficile colitis, which is really a minority of patients. But in the infant, when this gut microbiome is established, if you look at these numbers from the US, an average of almost 3 courses of antibiotics by the age of 3. Mothers get antibiotics during delivery to prevent staph infections afterwards, and 11 courses by the age of 10. Now, why is this timing important? Because it takes about 3 years before the gut microbiome is fully established. During the time it's plastic, developing, very sensitive to influences that can shape its direction. So all these influences that I showed here have a major influence early on in life. Once you're 5 or 10 years old, we're pretty resilient to any of these influences. Should also say the family environment. I've had several talks about foster situations. The family environment, the presence of pets, is also a very important factor that can be seen. So there's multiple things that go into this programming. Stress of the mother, which kind of overlaps with what we know about stress, the stress effects on, for example, the HbA axis, and I'll come back to this in a minute. So this influence of early environment factors on early brain development, this is important because it's not only the microbiome is being developed, but also the brain goes through major developmental processes during this early time period, longer obviously than the 3 years. But you have 2 phases of the brain-gut microbiome axis that are completely plastic during this early time period. Now the question is, do the microbes play a role in these processes? So this is a slide from the time, so we were involved, this is when I first met Michael Meany and had a meeting together in Sedona, also Sonia. So this model of maternal separation, an early life stress model that has been shown in elegant experiments to affect the HbA axis responsiveness, anxiety-like behavior, and the epigenetic mechanisms for that have been determined. It's the unique maternal behavior that is the determinant of the outcome of this early life programming. So the question is, do the microbes play a role in this process as well? It seems to be the case. So a study came out by a group in McMaster University, and make a long story short, in this study, the major findings were that maternal separation alters the HbA axis and colonic, the neural regulation of the colon in a way that was independent of the microbes. So it was also seen in animals that did not have their own microbes, so-called germ-free animals. But the development of anxiety-like behavior was not seen in these germ-free animals. So 2 components of that early life stress appear to be mediated by different mechanisms. One is dependent on the microbes. One is independent and has to do with this programming of the HbA axis through epigenetic processes. Even more intriguing is this story. And I should emphasize, whatever I tell you here are studies obtained in animal models, in mouse models. And you could criticize some of these models because there's always a comparison between animals with a normal gut microbiome and those that don't have one, the so-called germ-free animals. So that's not something we can do in humans. So this study showed that stress during a pregnant mother, fairly mild stress by chronic stress of a pregnant mouse mother, resulted in a change of the microbial composition of the vagina of the animal. So this decrease in vaginal lactobacilli. And that led in turn to a change in the gut microbial composition of the newborn animals. And you can see that this was different in females, the top 2 rows, and in males. So in females, there was a decrease in certain taxa of the microbes. You see this shortened bar. Some specific microbes were reduced. In males, they started out with lowered abundance of these same microbes, but the same situation increased their relative abundances. And they also showed that these changes in the early gut microbiome, this sort of fits together with what I told you about how the system is plastic in the beginning and how both microbes and brain are in process in development in the first 3 years. These changes were also associated with brain changes, early brain changes in these animals in terms of amino acids in different brain regions. So this is a fairly phenomenal thing, that the stress of the mother would be transmitted to the infant through a change in the vaginal microbiome, and that's the canal where the infants go through during birth. So there's clearly this early programming through this vaginal mechanism. It's not just the vagina; it's also the interaction of intestinal microbes and vaginal microbes in the birth canal area. If this plays a role in humans, we don't know, but it's certainly a very intriguing mechanism by which what I showed you on an earlier slide, this influence of stress to the mother or diet of the mother can be translated to the offspring through this change in the vaginal microbiome. So unfortunately, I don't have a pointer, so I just walk you through from the left. So there's our genes. This summarizes the factors that play a role in the development of the brain-gut microbiome axis. So there's the gene expression for brain proteins. Early adverse life events affect this by methylation processes. There's also the gene expression at the gut level, again influenced by epigenetic factors. The gut microbes early on in life have been shown to have an influence on stem cells in the gut, an epigenetic mechanism. And through both these influences, the psychosocial influences from the environment and the microbial influences, the system is programmed in a direction that the gut-brain microbes interact for the rest of the life. And ultimately, this programming results in certain networks within the brain that are important for different functions, like sensory motor network, salience network, emotional circuits. So for example, we have found in our studies in associations that a history of early adverse life events affects the sensory motor system in the brain, in the adult. But the same thing happens really at the gut level. So this early programming really sets us up with these networks, both at the gut and the brain level, which interact with each other and ultimately determine the clinical outcome. So rather than thinking about psychiatric or brain disorders as simply located within the brain, I think we have to take into account today, based primarily on animal studies, that this really happens both at the gut and the brain level, and they're both psychosocial factors as well as environmental factors, and that could be anything from, I mentioned earlier, the pets in the family, the antibiotic exposure, the close interactions, siblings in the family that have an influence on the microbial environment that this child grows up in. Why is this early programming important? So similar what has come out of the earlier work about the HPA axis programming. So we think that many developmental brain disorders, including autism, schizophrenia, depression, anxiety, really start early on during these first 3 years in life where this programming happens, and that the microbes do play some role. Needs to be determined in humans how big that role is compared to the brain, the direct brain factors. And so we know this, for example, for autism spectrum disorders, that elegant work from Sarkis Osmanian at Caltech, that has shown that the early microbes play a significant role in that treatment in this mouse model can have an influence on the clinical outcome if it's done early after birth. The other disorders, metabolic disorders like type 2 diabetes, metabolic syndrome, or the neurodegenerative disorders are probably not programmed early in life, but they involve, now it's pretty good evidence that nutrition and the microbes and influence of nutrition on the microbes play a role over a lifetime to induce some of these neurodegenerative processes. So what happens early in life, particularly important for the neurodevelopmental diseases. So take-home message, brain can influence the gut microbes, and gut microbial signaling molecules by multiple communication channels can affect the brain. And these interactions play a prominent role in brain development and programming. The establishment of the microbiome in early life is influenced by many factors, including the prenatal environment, mode of delivery, early nutrition, and perinatal stress. And early life gut microbial programming is likely to have a significant effect on brain development and several developmental brain disorders. So if you want to learn more about this and be updated on a regular basis, text this word mind-gut to the number on your cell phone, 66866. This is kind of a test. I've found that people under 30 are the best in actually doing this, and over 50 are intimidated what this is going to do. So it's a pretty young audience, so I think most people will be able to do this. And thanks a lot for your attention.